My name is Martine Pope, and I'm currently a rising junior at Cornell University. And my project is Cells Without Borders, optimizing oligofish in maize. So, a few basic things. What we were interested in studying was polyploidy, which is the doubling of genomic material within a cell. And how we were doing this is using FISH, which is fluorescence in situ hybridization. And that is using little pieces of DNA or RNA connected to a fluorophore as a probe to make it glow under fluorescence microscopy. And specifically, we're using oligofish, which uses libraries that cover the entire chromosome. As we can see here, we see hybridization in the third chromosome and hybridization in the first chromosome versus the entire staining of everything. And this is interesting to study because polyploidy is like the reason a lot of our most successful crops are so successful, such as cotton, peanuts, or wheat. However, new polyploids are often unsuccessful, and we're trying to understand why they're infertile or just like not very prolific. So a lot of my project specifically was optimizing this protocol so we could get a larger amount of images for our like future research. And the main ways we did this were through anther digestion, myocyte extraction, and cytoplasm removal. The cell wall in plants makes this procedure very difficult because it interferes with probe infiltration and also interferes with the removal of the cytoplasm. And the sort of like biggest improvement we made was using a droplet of acetic acid on over-digested anthers would allow us to remove the myocytes in a state where the cytoplasm was very easy to remove. And we increased the number of myocytes that were successfully imaged from about 4 per 4 slides to about 600 in only, or not 600, sorry, 206 slides. So we were able to get a lot more images, and those will be analyzed for non-homology and any sort of other chromosomal aberrations so we can better understand why a polyploid organism is successful versus not successful.
So do you think this would be useful with other crops plants outside of maize?
Yeah, I think so. A big challenge in, like I said, in any sort of crop plant is the cell wall. And I think our procedure is like very optimized for any, like specifically corn has a particularly difficult cell wall to remove. And that's why we were experiencing so many challenges, but this protocol could work for any sort of crop plant we want to study.
Great, thank you so much.